Thank you very much. So today we will have for our COSTU webinar, FredericFrottin. Just a few words about Frederic, and then I will leave Frederic to talk about the seminar. Frederic made his PhD with me in 2011. He was working at the time on the impact of a specific N-terminal protein modification on proteostasis, mainly in plants, but he was working also with translating all the experience in yeast, in archaea, and human cell. And immediately after his PhD, he joined Ulrich Hartl in Munich, and he started to work on different factors that were related with the protein quality control and neurodegenerative diseases. And it's there that Fred started to have this passion for nuclear proteostasis. And after a long postdoc in Munich, he could come back in France in 2020, I think, I don't remember. He got a CNRS position, and he came back with his own project where he tried to find a dialogue between condensate, nuclear quality control, and also protein modification. So I think I said everything, and I leave Fred to give the talk and overview about this fascinating project.
Okay, thanks. Thank you very much, Carmela, for this very kind introduction. So, yes, I am FredericFrottin, and now you know everything about me, my passion for nuclear proteostasis. You also know very well Carmela, so I'm working with her, and we also work on protein modifications, and for me, mostly for the proteostasis side, because these modifications are controlling also protein homeostasis. So it's a great pleasure to talk today because I have the opportunity to cover my interest in nuclear proteostasis. That's why today it will come in two flavors. I will first describe a pathway that we discovered quite some time ago now during my postdoc, which is a nuclear protein quality control pathway. And in another but related part, I will talk about nuclear toxicity pathway that are triggered by a specific mutations. And probably the shared, they intersect, both of them, at the nucleolus. So proteins, you know, of course, that they are essential workhorses of cellular functions, but they need to acquire their three-dimensional structure, a very specific structure, to perform their cellular task, and this is called the native conformations. But this conformation is often only marginally stable in the cellular environment. That is why cells tend to actually populate what we call misfolded species. And this is further enhanced by a range of endogenous and external conditions, such as, for example, crowding will enhance protein misfolding, but it could be mutations, aberrant translation products, modifications, and also, of course, a range of prototoxic stressors. And if cells fail to clear in a way or another these misfolded protein species, they can collapse and form protein aggregates. What I did not tell you so far is all these aberrant protein species, misfolded aggregates, and different kind of aggregates or even oligomers, they are thought to be toxic for cells. That is why cells from all kingdom of life invest, maybe I can take the pointer options, yes, invest in a vast network of components, so-called proteostasis network. In humans is nearly 3,000 components now that are here to remove, to eliminate the species, or at least to mitigate their toxicity. So beyond this component, cells also can make use of strategies actually to deal with this aberrant protein species. One example would be asymmetric cell divisions, where the mother cell will retain aggregated material within the cells, and then leaving the daughter cells with a healthy proteome. There's other strategies, and today we will dive into subcellular sequestration, of course. So I have to mention that so far the toxic species are still a matter of debate. I guess most species could be toxic in some conditions, and they can overtake the proteostasis network by different means. Another thing that is important is that protein aggregates, which are, of course, you know that for sure, that they are all related to various diseases, including many neurodegenerative disorders. These aggregates are found in various subcellular compartments, in particular in the nucleus, in the nuclear compartment. And you can see examples here for Huntington's diseases or ataxia type 1, ataxin 1 aggregate within the nucleus. Or again, in amyotrophic lateral sclerosis, you can see some aggregates in the nucleus. However, protein quality control pathway and how the nuclear environment deals with aberrant nuclear protein is not so well known as compared to the situation in the cytoplasm, for example. So a few words about the nuclear compartment. So it is, of course, a very specific proteome enriched in metastable proteins. That means it's very sensitive to stress, it's highly vulnerable. That's why there must be some pathway and strategies to basically deal with this proteome. A feature, of course, of the nuclear proteostasis network is that it has to be adapted for conformational maintenance, because there is no protein biogenesis in the nucleus. That means de novo folding is not required within the nucleus because protein enter the nucleus already folded. And of course, this specific organelle takes care of a lot of very important essential cellular functions. And most of these functions make use actually of what we call membraneless organelle or biocondensates that arise through at least liquid-liquid phase separation phenomenon. So the largest of these biocondensates in the nucleus and in cells actually is the nucleolus. So the nucleolus is well known for being the site of ribonucleoprotein particle biogenesis, especially the ribosome, of course. So the nucleolus is also a very specific biocondensate because it contains several subdomains. There is 3, but in fact now I think we can count up to 5 different domains. So these domains do not have a membrane, but they do not nonetheless intermix with each other because of differential surface tension. And these various domains actually correspond to various steps of the vectorial ribosome biogenesis. So then we wanted to, of course, when we started this project back in Munich, to have more information about how the nucleus will deal with misfolded proteins. And for that, we make use of firefly luciferase. It's a protein model, the labile proteins, and we fused it to the thermostable GFP. And of course, we targeted to the nucleus, as you can see here, with a regular NLS sequence. And then we express this protein in HEK cells. So upon heat shock, it's 2 hours at 43 degrees. What we observed is basically a relocation of this protein inside the nucleolus, as seen by the colocalization with the nucleolar marker nucleophosmin, NPM1, here in red. And this relocation within the nucleolus was dependent on misfolding, because if you treat cells with the small molecule PBT, which is a luciferin analog, that will bind to the active site of luciferase and stabilize its fold, and this is acting as a chemical chaperone. Basically, then the protein will be stabilized, and as you can see, during the stress, there is no more relocation of luciferase within the nucleolus. So upon recovery from stress, basically the nucleolus, the nucleolar misfolded luciferase will exit the nucleolus. So of course, I just told you a few minutes ago that the nucleolus, a lot of various compartments, so we wanted to have more information about the precise localization of misfolded luciferase within the nucleolus, and we used through collaboration DNA paint-based super resolution imaging, and we could identify that our misfolded protein model accumulate in the NPM1 positive compartment, which is called the granular component, the most external part of the nucleolus, and does not accumulate in these two other parts of the nucleolus, the fibrillar center and the dense fibrillar component. Okay, so of course, when we talk about stress, we have to look at HSP70, and actually it's known since decades, thanks to seminal work from Susan Lindquist, for example, that HSP70 in stress condition translocate in the nucleus and can also accumulate in the nucleolus. And we actually did see that as well. We can see that HSP70 colocalizes very well with our luciferase model protein within the nucleolus. And what was quite nice to see is this was HSP70 could still enter the nucleolus upon stress, even when luciferase fold was stabilized with the chemical chaperone, which told us that HSP70 enters the nucleolus either as a free form or together with endogenous substrate. And like for luciferase, during the recovery, actually, HSP70 goes back to the nucleus, the nucleoplasms, and the cytoplasm. And we could identify by inhibiting HSP70 ATPase activity here that HSP70 activity was required for the refolding, but also the repartitions of luciferase from the nucleolus to the nucleoplasm during the recovery. And actually, HSP70 in this condition stayed somewhat sequestered at the nucleolus with our model protein, firefly luciferase. So then, of course, we wanted to have more information about luciferase within the nucleolus upon stress. But for that, we needed a better tool. That's why we altered our firefly luciferase. We added a nucleolar targeting sequence so that our luciferase could be within the nucleolus, also in control conditions for comparison mean. And we could do some FRAP experiments, and we identified that actually upon heat stress, upon misfolding, luciferase was largely immobilized or at a very low mobility in the nucleolus upon stress, and we could confirm this was dependent on luciferase misfolding using the chemical chaperone and a bunch of other control. I'm not showing also today for time consideration that actually luciferase, misfolded luciferase, and misfolded protein immobilized the mobility of nucleophosmin and other nucleolar protein from the granular component phase of the nucleolus. And indeed, when we did immunoprecipitation experiment of luciferase, regardless if it's a nuclear luciferase that translocates into the nucleolus upon stress, or the protein which is always in the nucleolus, we could see that luciferase specifically interacted with nucleophosmin when it was misfolded. So then this previous experiments told us that there should be a pathway, an endogenous pathway of accumulation of protein within the nucleolus upon stress. That was not only true for our favorite model protein. And for that, we did interactomic of GFP and PM1. And we did that in several conditions, and we could indeed identify that about 200 proteins reversibly interacted with this GFP NPM1, as you can see here. And also these proteins, even a little more, needed the ATPase activity of HSP70 to exit the nucleolus during the recovery. You have here one example of such protein that we find is BRD2, bromodomain containing 2, that reversibly translocate within the nucleolus upon stress. Okay, so then we wanted to, of course, try to understand, to investigate the functional relevance of this insertion or accumulation of luciferase within this phase-separated compartment. And for that, we had a very simple idea, is to destroy basically the nucleolus, and we can somehow do that using actinomycin D. So actinomycin D will inhibit the ribosomal RNA synthesis, and this will lead to the disassembly of the nucleolus, as you can see by the distribution here throughout the nucleoplasms of the nucleolar marker, nuclear phosmin. And in these conditions, upon stress, we had the formation of nucleoplasmic foci. And these foci, they were extremely bright, and we could measure it. They are actually much brighter, and the local concentration of luciferase within this foci is 3 times higher than the luciferase within the nucleolus. And more importantly, actually during the recovery, the cells fail somehow to remove this foci, this nucleoplasmic puncta of luciferase, as comparison to luciferase that were in the nucleolus. So which tells us that having a nucleolar environment organized as a liquid-like phase promote the efficient release of misfolded protein during the recovery after the stress. And in fact, what we observed is we can stain our cells with dyes that will recognize amyloid features. And with nucleoplasmic foci of luciferase, we could see actually that these foci were positive for these dyes, which is not the case, or very little, when we have organized nucleoli. So even more, actually the nucleolus prevents amyloid-like protein aggregations, or at least delays it. So then we also wanted to probe another capacity, protein quality control capacity of this nucleolar phase. We wanted to see if this phase could be protective. But for that, we needed to switch our protein models, because firefly luciferase is not toxic per se. So we used another of models that we like very much, which are the so-called beta sheet proteins. So these proteins basically are alternating pattern of polar and non-polar amino acids. They form beta sheets, and they basically aggregate. This is a pure gain of toxic function proteins. And we have different variants of these proteins. We have an NLS variant that naturally actually accumulates within the granular component phase of the nucleolus. You can see super resolution imaging here of the cells. You can see it colocalizes very well with the granular component marker nuclear phosmin. And we also switched actually this NLS for another kind of NLS called PY. And in this case, the beta sheet protein accumulated within the nucleus, but now formed puncta structure within the nucleoplasm. So basically, we could then compare the effect of these two proteins on the viability of the cells, and we could see that when the beta sheet proteins are within the granular component phase of the nucleolus, it is more protective than when it's outside in the nucleoplasm. So this means that the accumulation within the nucleolus reduces the toxicity of such amyloid-forming protein. So then we basically used our two model proteins. We co-expressed these two proteins in HEG cells, and we could observe that our luciferase co-aggregated with nucleoplasmic foci of beta sheet proteins, and the nucleolar protein quality control pathway could work somehow. You could see the translocation within the nucleolus of firefly luciferase that could actually be eliminated then during the recovery. Then if we switch our nucleoplasmic beta sheet proteins for the beta sheet proteins that accumulate within the nucleolus, what we observed was actually quite fascinating for us. We could observe that the luciferase could accumulate upon stress within the nucleolus without any problem, and then repartition back into the nucleoplasms during the recovery. And this was quite extraordinary because within the same phase, the granular phase, you have two proteins that are extremely aggregation prone, and somehow they do not co-aggregate, or at least not in an irreversible manner. And this is some sort of all days capacity of the granular component phase of the nucleolus. So far I told you that the nucleolus can only be good for you, but we still identified some limitations. So basically upon prolonged stress, we observed that first nucleoli get bigger because of the influx of misfolded proteins, and with time it loses its round shapes, spherical shapes that is more consistent with liquid-like features, but you start to see sharp edges. And in fact, the amount of immobilized NPM1 is larger and larger. And also we could see with this amyloid recognizing dyes, MET, we had developing staining with time under heat shock. And the consequence of that is a loss in protein quality control capacity, because you see that during the recovery, cells have more difficulties to eliminate nucleolar luciferase when cells were upon prolonged heat stress. So what I showed you so far as a first stop is this pathway where misfolded protein accumulate transiently within the nucleolus stress. So this is a clear subcellular sequestration. It has another function in protein quality control than only sequestering protein. It shields also reactive and toxic surfaces, as we have seen with the beta sheet proteins. It has also a chaperone-like function, which is highly related to its liquid-like features. And by holding protein in a state compatible for further processing, such as refolding, it also prevents amyloid-like and irreversible protein aggregation. So I didn't talk so much about it, but it's somewhat a one-pot reaction, the nucleolus during stress. Lot of protein quality control component would immerse or migrate towards the nucleolus. I showed you HSP70, but there's a bunch of other chaperone and protein quality control components, and we still don't really know what they are exactly doing there. So let's talk now about this C9orf72 mutation. So this is an hexanucleotide repeat expansion. It is in patients, mostly amyotrophic lateral sclerosis and frontotemporal dementia patients. So these mutations is the most frequent familial cause of these diseases. This was discovered, as you can see, not very long time ago. So this is an expansion of this repeat, this motif, G4C2, and patient could be several thousands of these repeats. And in 2013, it was identified that these repeats are translated by unconventional translation, because it does not depend on an ATG, basically. So it is translated in all directions, sense and antisense, and in all frames. And since it's an hexanucleotide repeat expansion, this gives rise to dipeptide protein repeat. So actually there are 5 different DPRs which are made by this expansion. The most abundant in patients seems to be poly-GA. So of course, people are very interested in understanding the toxicity induced by these mutations. So the regular explanation, of course, is first the loss of function because C9orf72 protein seems to be involved in autophagic processes. And for the gain of toxic functions, there's, of course, the DPR toxicity that I explained to you, but also the RNA toxicity because the G4C2 containing RNA are thought to form actually structures, G-quadruplex structure within the nucleoplasms and sequester, for example, RNA binding proteins away from their cellular task. So we were at that time interested in DPRs because also they, as you can see here, the poly-GA can accumulate also in the nucleolus, and we wanted to have information about compartment-specific toxicity pathway induced by these proteins. Beyond that, we also wanted to somehow sort out the contribution of the potential RNA G4C2 toxicity than from the protein toxicity. And I can tell you that it's not an easy task. So for that, we engineered poly-GA, so GA 65 repeats, and we fused it to GFP, but the GA is expressed from a degenerated sequence. So this is the sequence we used, and it is there. It does not contain a single G4C2 motif. And we also compared that to proteins, poly-GA, of very similar length of the repeat, 73, but this time the GA are made from purely G4C2 constructs. So both constructs are driven by an ATG, and we also added signal sequence, NLS and NES, to sort out also the compartment-specific effect that we could see. So first, let's focus on the protein-mediated effect. So basically, the proteins, when we express it in X cells, it aggregates very quickly, very strongly. It aggregates in their respective compartments, untargeted variants aggregates mostly, I would say, in the cytoplasm, but also in the nucleus. The NES forms are, of course, in the cytoplasms. And we have the NLS variant that accumulates in the nucleus. I am not showing all the time all the controls, but we have a bunch of various NLS sequences and their inactive mutants to control all our experiments. So under the microscope, the aggregates may look similar, but in fact, they are different in terms of solubility. Using the classic filter trap assay, we could identify that these nuclear variants, these nuclear proteins, were always somewhat more soluble than their cytoplasmic counterparts. Beyond the solubility, actually, the nuclear variants were inducing toxicity as compared to their cytoplasmic variant that were not producing much toxicity in our hands, even for a similar expression level. We did all our control, and each time we had a construct that accumulated the poly-GA within the nucleus, it was toxic in our hands. And this was quite striking, actually. So, of course, we wanted to have information about the potential localization of this nuclear aggregate inside the nucleus, where it would accumulate. So actually, they are not within the nucleolus. We could see that, but we always observed that the nucleolus was somewhat altered by the presence of this nuclear construct. It is like this granular component phase of the nucleolus dissolved by the presence of these aggregates. So far, we still don't know how this mechanisms occurs, but it's something we always observed, and we did not observe that in presence of cytoplasmic poly-GA aggregates. So since we could see an effect on the nucleolus, we wondered whether this could have an impact on the nucleolar protein quality control pathways that I described earlier. So we use this time basically a red variant of our NLS luciferase. And here it's a control condition, so it's co-expressing GFP only, and you can see that the pathway is working very efficiently. Luciferase can translocate upon stress within the nucleolus and repartition back into the nucleoplasm during the recovery. And then when we co-expressed, instead of GFP, our nuclear proteins, basically we see quite strongly that luciferase enters the nucleolus, but somehow failed to be removed to repartition back into the nucleoplasms during the recovery. So of course we didn't see this effect with the NES, with the cytoplasmic protein aggregates, and here you can see the quantifications. You can see that the recovery is much slower, much more difficult when cells express the nuclear GA protein. So just quickly, I also wanted to show you another experiment that we did for the first story, actually, is to use another DPR, an arginine containing DPR, PRs, because it is known that they accumulate quite strongly in the nucleolus, in the granular component phase of the nucleolus, and also interfere with other membraneless organelles. And indeed, it's what we also could observe. You can see here the PRGFP. We could measure that this protein immobilized a fraction of the nucleophosmin, and this had an impact on the nucleolar protein quality control pathway. You can see that luciferase stays somehow trapped within this granular component phase in presence of the arginine-containing DPR. And here it's a control with a nucleolar GFP only that is working very fine. So here we have basically another situation where the DPR is inside the nucleolus and negatively impacts the nucleolar protein quality control pathway. So then it's time maybe to add the G4C2 containing constructs. So these are constructs, as you can imagine, that are difficult to actually work with. You can easily spot this construct in cells if you do a FISH experiment and you probe your cells with a C4G2 probe. You can see this mRNA puncta inside the nucleus. In terms of protein expression, is relatively similar to protein made from the degenerated sequences, and also the aggregation and the targeting sequences are working, of course, in the similar way. So we actually tested whether this construct will have an impact on mRNA, and we did that also by FISH using the PolyDT probe. And this is what you expect from normal cells, happy cells. You have only this little mRNA puncta and a lot of cytoplasmic mRNA, of course. And what we clearly observed is each time we transfected our cells with constructs that were containing the G4C2 repeat, we had a massive accumulation, retention of a total mRNA within the nucleus. This was, of course, independent on the targeting signal. And as you can see also with the degenerated construct, this is also independent of the proteins, because regardless if you have a cytoplasmic or nuclear aggregates that are not made from G4C2 construct, we don't have at all this accumulation of mRNA. It's like the anti-vector control, basically. So then, of course, we wonder if it had an effect on protein translation. So for that, we measured protein translation using the well-known pyromycin derivatives, OPP, and we pulse label our cells with these molecules. And after cell fixations and click chemistry, we could assess the efficiency of protein translation. As a control, you can see, for example, that treating your cells with the cycloheximide inhibitor, the cycloheximide protein translation inhibitors, you don't have any staining, basically. So what we observed is that at the protein level, when you have a cytoplasmic GA aggregate, this has no effect as compared to empty vector or GFP expressing cells. So then when you looked at cells with GA aggregates inside the nucleus, you see quite a strong decrease here in protein biosynthesis. And you can see it here on the pictures. You can see the surrounding cells don't have any expressions, and in the middle you have like this sharp decrease in staining intensity, which indicates a much reduced translation efficiency. And then if you looked at proteins that are expressed from G4C2 containing DNAs, you can see indeed very strong also decrease in the efficiency of translation. Quite interestingly, I would say, we saw some sort of synergistic effects of the proteins, the NLS-GA and the G4C2, because we have even stronger reduction in translations when we had the NLS G4C2 expressed, probably because, or maybe because two different pathways are targeted by this construct. Maybe the NLS-GA that impairs the nucleolus, probably the ribosome myogenesis is altered. This remains to be formally demonstrated, of course. And the G4C2 will alter the mRNA, of course, pattern in cells. So then it is not surprising that the G4C2 containing construct induced a strong toxicity on transfected cells. As I showed earlier, of course, the NLS-GA is also toxic in cells. And then at that time, we also were concerned that the G4C2 toxicity that we were observing was not due to the G4C2 mRNA, but would be due to RAN translation product, especially the arginine-containing proteins in the DPRs, because these arginine-containing DPRs were suggested to be toxic in some cellular models. So that's why we engineered GR and PR, also 73 repeats, matching length, and express it in cells. They behave as expected. They accumulate mostly in the nucleolus. But in our end, actually, they were not toxic, as you can see here. And on Western blot, we could detect them, of course, for PR here and GR. And for our G4C2 construct, we did not detect specific staining for PR or GR, as you can see here. That's how we could rule out the potential toxicity of this G4C2 that could be due by this kind of RAN translation product. So this suggests this is most probably due to the G4C2 RNA itself. So let's summarize everything. So basically, I showed you that the nucleolar protein quality control pathway could be altered by poly-PR, which infiltrates, immerses into the nucleolus and disturb its liquid-like properties. We have a similar consequence induced by poly-GA protein inside the nucleus. Also, it is not inside the nucleolus. It alters the nucleolar structure as well. And the result is a loss of protein quality control of this pathway, of course. G4C2 RNA also, of course, has a strong impact on mRNA, on protein biogenesis. Also, nuclear poly-GA has an impact on protein biogenesis, maybe due because of reduced ribosome biogenesis. This has to be demonstrated, of course. I didn't talk about it today, but we did a lot of other work on the cytoplasmic GA proteins and other disease-related protein aggregates that you can check if you're interested in. And now, as a perspective, a few words, basically, we are still continuing to to investigate these two aspects of nuclear protein quality control, and the pathways as well as the toxic protein aggregations. As Carmela mentions, I'm also interested in protein modification because they can basically alter the capacity to phase separate, because it will alter the interaction strength or the valence of a protein. So basically, we are working on finding regulators of this pathway, and we try to also understand at a molecular level what's happening inside this nucleolar phase with this chaperone-like capacity and the misfolded proteins. And we are also now working with nuclear aberrant proteins and try to understand how they can impact nuclear function and basically cell survival. So I have to thank, of course, many people. So there's a lot of work that's been done. All the work has been done at the Max Planck Institute of Biochemistry with a lot of good people, actually. And now this work is continuing within Carmela's lab, and I would like to highlight the contribution, of course, of Julia. She's working now on the nucleolar protein quality control pathway, and Mathieu that is working on nuclear aggregates. I thank you for your attention as well, and I'll be happy to answer to your questions, comments, ideas.
Thank you, Freda.